Hello everybody. This is an experiment for me because I've never recorded a talk before in my life, and I wish I was with you live and in colour myself, but it may be some time before I get back to Szeged, a city of which I have very fond memories. So here goes. I'm going to tell you the story of my life, that is to say my scientific life. And starting at the beginning, I started to do my PhD in a lab in Cambridge in the biochemistry department in 1964. And my supervisor, I said, what am I going to work on? And he said, well, Tim, why don't you go to the library and find yourself a project? So I went to the library, and after about a week I found a very nice project, which I think fortunately for me absolutely did not work. So for the first 6 months or so of my PhD, absolutely nothing happened, except that I sort of learnt my way around the lab, which was okay. And the key moment really where my scientific career could be said to start was when I went to my first scientific meeting. It was only about a quarter of a mile away in the chemistry department, and it was a meeting devoted to hemoglobin, the stuff that makes your blood red and carries the oxygen around your veins. And I heard two talks there that made a deep impression on me, and in fact set almost I could say the course of my research for the rest of my life. So the first talk was by a man called Henry Borsuk, who came from Caltech, and he'd worked on two very unrelated things. One was sea urchin eggs, and the other was the role of iron in hemoglobin synthesis. Heme, that carries the oxygen, contains iron, and it turned out that if you starve the cells that are making hemoglobin of iron, they stop making the protein globin. So that was the first talk. He also introduced me to sea urchin eggs, and sea urchin eggs, he described, make almost no proteins when they're just sitting there unfertilized, but the minute they're fertilized they activate protein synthesis, and that's very important even for the very first division. They don't go anywhere unless they can make new proteins. That sort of made sense. And the second talk was by a man called Vernon Ingram, and he was also studying the question of the coordination of heme and globin synthesis. And I think it'd be a good idea to show you what that looks like. So here is John Kendrew looking at his model of myoglobin, and as you can see, the protein curls its way around and makes a little fold where the heme slots in. And Vernon's problem basically was how does the heme, when does the heme go in, and how does it control protein synthesis? And he presented some very interesting data which suggested that the ribosomes which are making the protein globin go along the messenger RNA until they reach the place where the heme should come in. And if there is no heme, they simply wait there and form a queue behind the service point where the heme ought to be inserted until the heme comes in. If the heme comes in, then they can go on and finish making the chain. So this was a remarkable mechanism, very easy to understand, or difficult to envisage how it might actually work. And I went back to tell my friends in the lab about this amazing talk that I'd heard, and my colleagues pointed out that actually the evidence presented exactly contradicted his own theory. He'd misinterpreted his data, basically. And so we decided, and this is something I really can't understand how we had the courage to do it, we decided to check it out for ourselves. And when I say we, there were really two key people. You'll see that this talk is really mostly about people. The first was on the right, Lou Reichardt, who is a very distinguished neurobiologist these days, but he started out his PhD in Cambridge and he shared the bay with me, and he in fact was trying to study the coordination of heme and globin synthesis, and like me with his first project, failed completely. I don't know why. I think it may be heme is rather difficult to dissolve, and maybe he didn't succeed in dissolving it. And the other person on the left is Tony Hunter, and in the background are reticulocytes, the cells that actually make hemoglobin. And later on it turned out that it's very easy to make a cell-free preparation simply by suspending the cells that are making hemoglobin in water, distilled water, which bursts open the cells by osmosis, and you get a beautiful deep red supernatant, which will make hemoglobin, even though there are no more cells involved. You have to add a source of ATP, an energy generating system. But that's all you have to do, and it works extremely well. And you can buy this as a kit. We used to make our own. In those days, you couldn't buy it. But Lou knew how to make it, and that was great. So off we set. Tony Hunter and I really did our PhD together, and we found that actually the ribosomes did not form queues. The whole thing was a complete nonsense. And then a very important thing happened. I went to my second scientific meeting. This was in the spring of 1966, and it wasn't a quarter of a mile down the road. It was in northern Greece. And there I ran into a man who became a great friend and mentor to me, and I actually spent the summer in his lab in New York and then went back to do a postdoc. And on the right-hand side is shown the kind of thing that we could do with this cell-free preparation. That is to say, we took samples at timed intervals and measured protein synthesis. And as you can see, if you add heme, protein synthesis carries on at a nice high rate. In fact, it's pretty much the same rate as they would have been if you hadn't broken the cells open. But if you leave out the heme, then protein synthesis starts off all right. You make a little bit of globin, and then it suddenly cuts out and goes to a new low level, typically 10% or something like that. If you add back the heme, it starts up again. So we have a reversible control mechanism, and we would love to understand what that control mechanism is. So I spent about 2.5 years in New York trying to work it out, but actually it was deeply unsuccessful because we didn't know enough about how protein synthesis was controlled at the time. We really were completely ignorant, and we kept on doing this same experiment, which always worked. It was extremely reproducible, but it didn't lead anywhere to any sort of fundamental understanding of the problem. And I did find out one or two of things while I was a postdoc, but they were sort of secret projects at night. And the most important one was done with a collaborator in the cell biology department at Einstein. One of the nice things about Einstein was it was very easy to collaborate, and people liked to do it. And so I found somebody who was working on poliovirus, and we discovered that tiny, tiny amounts of double-stranded RNA, which is the replicative form of poliovirus, inhibited protein synthesis in these reticulocyte lysates in exactly the same way that leaving out the heme did. And that was very, very strange, because chemically there's absolutely nothing in common between double-stranded RNA and heme. And remarkably, perhaps the most important thing was that when you worked out how much double-stranded RNA would inhibit synthesis by how many ribosomes, it turned out that one double-stranded RNA molecule could inhibit something like 10 million ribosomes. And that immediately said that whatever this inhibition was, it must be catalytic, because the trigger is vastly amplified when it comes to its final effect. But we had no idea how it worked. And so after that, I went back to Cambridge, where I joined forces with a man who died recently, he's a couple of years older than me, called Richard Jackson. And Jim Watson, in one of his books, says that one of the great tricks about being a good scientist is always to work with somebody who's cleverer than you are. And I think that's excellent advice, and that's what I did. Richard was an extremely deep and careful thinker. I was a little more slapdash, and between us we made, I think, a very good team. And it was we worked together for about 20 years, in fact, and it was an enormous pleasure, the two of us together. So what did we find? Well, we didn't find this. Somebody else discovered the fact that when you left out the heme, what happened was that an inhibitor of protein synthesis formed, and that inhibitor stopped ribosomes translating the messenger RNA. In fact, they stopped the ribosomes from going on to the messenger RNA in the first place. So all the polysomes, which are messenger RNA covered with ribosomes, disappeared, and you got a huge increase in single ribosomes unattached to messenger RNA, not making globin. Now, while I was in America, Richard and Tony Hunter, who I've already mentioned, discovered a very important thing, which is the way you start protein synthesis is with a special initiator tRNA carrying methionine. And so we started looking, using methionine as a label. It's a very nice label because S35 is nice and radioactive, and it's cheap and easy to make. And actually, we at one point actually made our own. And a graduate student called Chris Darnborough discovered that when you left out the heme, or when you added double-stranded RNA, there was a mysterious complex, which wasn't a full ribosome, it was half the ribosome, went away. And that complex went away before protein synthesis itself shut off. So that we knew that this complex was probably a complex of initiator tRNA, which then led us to realize that what happened, the way you start up protein synthesis, is by the small ribosomal subunit getting charged with initiator tRNA, and that binds to the messenger RNAs and goes along looking for the start of the messenger RNA, looking for the first methionine codon in the messenger RNA. And if you can't form that complex, of course, you can't start protein synthesis. And so the disappearance of this complex provided a very nice reason for why protein synthesis stopped. But we had a hard time publishing it because everybody knew that transfer RNA only binds to messenger RNA when the messenger RNA instructs it. The idea that the tRNA anticodon could select the start of the messenger RNA was completely the wrong way round for everybody. I mean, in other words, they said that we didn't understand anything about how protein synthesis worked, but actually we understood it better. And it's always been my experience that if you find out something that goes against the current dogma, you always have a much harder time publishing it, and you better be very sure that you're right. So that's a good thing. Anyway, so that was, in a way, the first really, really important result that I shared in with my colleagues. And then a very fortunate thing happened, which was that the lab burned down. And I got a phone call at about 7 o'clock on a Saturday morning from Richard saying, "Tim, the lab has burnt down. Don't bother to come in because there's nothing left." So, of course, I immediately got dressed and rushed in. And indeed, as this photograph shows, the lab had been completely destroyed by fire, except that the liquid nitrogen refrigerator where we kept our most precious samples was still venting nitrogen, which meant that the interior was still at minus 100 and whatever it is, 180 degrees Celsius. And so the precious samples were still safe, and all we had to do was find a new refrigerator which hadn't blown its seal, and that was very fortunate, so that we were able to get back and in business in about 6 weeks. We had a wonderful story to tell. The lab has burned down. Can you get us a new this or new that and a new the other? And we had a new lab to go to. It was a teaching lab in the hematology department, opposite the famous laboratory of molecular biology, where they have, I think, 20, 25 Nobel laureates sitting there. So it was a really wonderful opportunity, and actually at the time we were deeply puzzled. We had lots of sort of results that didn't really make much sense, and it was really rather a good thing to burn them all up and have no possibility of consulting what they were. We sort of, it was good. And more importantly, we had new neighbors, new labs, all new equipment, thanks to the fire. And Max Perutz, who was then the director of the lab, was extremely kind and said we could use his stores and perhaps more importantly, use his tea room. And the tea room was full of the great and the good of modern molecular biology. And our hero at the time, I suppose, my generation, was Francis Crick, who seemed to know and understand everything. He left Cambridge about a year or two later, much to everyone's sorrow. But not only was there Francis Crick there was also Fred Sanger, who won 2 Nobel Prizes and figured out how to sequence DNA, Max Perutz himself, who worked out the structure of hemoglobin, Sidney Brenner. And not only were there these great pioneers, but there were also a lot of youngsters, people like me, who would become in future Nobel laureates. And the interesting thing about that was that we didn't really consider ourselves to be in anything like the same league as these giants of molecular biology. We just sort of were studying interesting things and were interested in what each other were doing, made suggestions, made criticisms. And in that sense, sort of science is really very easy. All you have to do is to find a good problem and work on it and put one foot in front of the other and you solve it. But you know, actually the reality is it's very, very slow, and usually things don't work, and most of your time is spent troubleshooting. But it was a very stimulating environment. We were rather sorry when the old lab was rebuilt and we had to go back downtown again. But in the course of doing that, and I think it was largely as a result of making this move, we actually solved the problem and discovered this inhibitor of protein synthesis was an enzyme called a protein kinase, an enzyme that sticks phosphates onto other proteins. And the protein that it stuck a phosphate onto was an initiation factor, the initiation factor that indeed bound Initiator Trna to the small ribosomal subunit. So that was very good, very satisfactory, and we sort of thought in retrospect why were we so slow to think of that as a possibility. And we found that not only did leaving out the heme cause the phosphorylation of this initiation factor, but also adding minute amounts of double-stranded RNA had the same effect. And we now know, actually, after many more years of detailed research, that there are 4 of these enzymes, all of them sensitive to different things. So there's the heme-regulated protein kinase, there's the double-stranded RNA regulated protein kinase, which actually is a very important part of the antiviral response. There's something that responds to starvation for amino acids. And there's something called the unfolded protein response, which responds to when a cell can't fold up the proteins it's making rapidly enough, it switches off protein synthesis so that the folding can catch up. But that was way in the future, and we weren't to know that. So, but an interesting thing now happened, which was that in a way we've solved our problem, so we don't have a problem to work on anymore. So what do you do? And I remembered that talk from Borsuk about the sea urchin eggs, and I wondered what was known about the control of protein synthesis in sea urchin eggs. So I invited Tom Humphreys to a meeting that we organized, which was devoted to the control of protein synthesis in Cambridge. And Tom came all the way from Hawaii, and it turned out, I didn't know this before because I'd never met him, that he was a very keen cyclist and he wanted to rent a bicycle. And in those days you couldn't rent a bicycle, so I simply lent him my bicycle, and on that basis we became friends. And what I also didn't know was that he was the director of a course in Woods Hole, the embryology course, and he said, would I like to come and teach next year in the embryology course in Woods Hole, and maybe we could do some experiments together on the control of protein synthesis in sea urchin eggs. So I leapt at the chance, and next summer indeed I went to Woods Hole and we started working on sea urchin eggs. We didn't find out very much that first summer, but I did find out how to get sea urchin eggs, and I learned that you can't easily tell male sea urchins from female sea urchins because all they do is shed their gametes into the sea, and they don't need to get together in order to fertilize each other's eggs. But there are males and females, roughly equal numbers, and you can find out which is which by stimulating them with a little electric shock, and that causes them to ejaculate their sperm if they're male, or lay their eggs if they're female. And if you mix the two together, this movie shows what happens after fertilization, namely that after about an hour the fertilized egg divides in 2. It's really very beautiful. And I was fascinated by that, and also learned that if you inhibit protein synthesis, they don't divide in two. They will synthesize the first round of DNA, but they fail to divide. And it was true what Borsuk had said. There was a big increase in the rate of protein synthesis after fertilization. So this is pretty interesting. So at least one or more of the newly made proteins must be involved in catalyzing cell division. But we made no progress whatsoever in any of these things that first summer, and my recollection is we spent an awful lot of time chatting in the pub late at night and sometimes going dancing. But the second summer I went back, now much better armed with the things you needed to do proper experiments in a funny place like Woods Hole, I ran into two other people, Eric Rosenthal and Joan Ruderman, and they'd been working on clams and discovered that when clam eggs are fertilized, there's no big increase in the rate of protein synthesis, but more importantly, the clam eggs start making new proteins. And this slide shows that new proteins A, B, and C are synthesized very strongly after fertilization. And another amazing thing happens after the clam eggs are fertilized. They undergo a huge change in their structure. The nucleus, which is very prominent in the unfertilized eggs, breaks down and they form the first meiotic spindle, which is then followed by the second meiotic spindle. And again, if you inhibit protein synthesis, things start going wrong, and the clam eggs also don't divide. So I began to be interested in the relationship between protein synthesis, new protein synthesis, and cell division, because we didn't know what proteins A, B, and C, the newly synthesized ones, did. And, but they were so prominent that it rather looked like they were probably rather important. And that same summer when we were studying A, B, and C in clams, there was a seminar from a man called John Gerhardt, a very distinguished enzymologist who'd now become interested in something else, which was the maturation of frog eggs. Actually, the maturation of frog oocytes. Now, a frog oocyte is the cell inside a female frog that will become an egg when she lays it. And actually, the maturation is something that normally happens in response to the sex hormone progesterone. And it takes place over the space of a few hours as the oocytes go down the oviduct to emerge outside the female frog to get fertilized by the male frog, who is generally clasping the female frog. And they don't really ejaculate, they kind of dribble their sperm over the eggs as they come out. And again, the movie shows that when you fertilize these, when you add progesterone to these oocytes, a white spot forms in the middle of the black part. And if you look underneath the white spot, what you find is that there is a second meiotic spindle with chromosomes lined up on this array of microtubules. And what Gerhardt described was the existence of this interesting factor called maturation promoting factor, usually known as MPF, which appeared to be the substance that actually is the cause of this change in the oocytes, and the nucleus again breaks down and this spindle forms. And a Japanese researcher called Yoshio Mazui had discovered the existence of MPF by a very simple assay. He took a mature egg with its spindle and injected that back into an oocyte, and the oocyte responded very rapidly now by undergoing maturation, and didn't require any new protein synthesis, whereas the first maturation triggered by progesterone did. So there were again sort of correlations between protein synthesis and cell cycle transitions. And Gerhardt had been trying to purify MPF to find out what it was, and what he discovered was that it was very unstable stuff, very difficult to get hold of, and he really didn't know what it was. But it looked like it was important. And since he gave that talk, several other facts emerged about it. which was that not only was this found in frog eggs when they matured, but it was also found in starfish oocytes, and later on in yeast, and indeed even in human cells. So this was a substance which apparently would trigger entry into meiosis or mitosis in any cell that you cared to inject it into, and it looked like it was the enzyme that catalyzed cell division. So I thought this was a wonderful project, and began to think about that and the correlation between protein synthesis and cell division. And later on, Gerhardt did, together with Mark Kirschner and his wonderful technician Mike Wu, who I met one summer in Berkeley, they discovered that MPF levels were always high when the cells were dividing and always low in between when they were synthesizing DNA, replicating their genomes. And as I say, this wasn't just something confined to frogs. You found it all over. So I began to think, and we began to wonder, can you detect MPF in sea urchin eggs by any chance? And we even discussed the possibility of doing experiments to detect that, and never carried them out. Sea urchin eggs are very much more difficult. They're much smaller than frog eggs, so never got around to it. But then 2 or 3 summers later, I did an experiment which gave the game away, and it's shown on this slide here. This is actually the second experiment, which worked a little bit better than the first experiment. And it's a very simple experiment. You just add S35 methionine to sea urchin eggs and fertilize them and take samples every 10 minutes. Now, I was looking for something completely different. I was looking for changes in the pattern of protein synthesis between properly fertilized eggs and parthenogenetically activated eggs. You can get sea urchin eggs to develop just by adding a calcium ionophore, dilute soap solutions, and various other tricks that have been discovered many years before. And I was curious about whether properly fertilized eggs made different proteins from these artificially activated eggs. I did the experiment, and to my astonishment saw that the most abundantly synthesized protein early on after fertilization disappeared just before the eggs divided. We later found out that it actually disappears just before the chromosomes come apart at the culmination of mitosis. And then it came back again, and then it disappeared again. And this was quite remarkable. And later that summer we had a look in clam eggs, and we found, and it's much easier to see in this slide here, that the proteins A and B, which are both translationally regulated, are these proteins which I call cyclins, because number one, I was a keen cyclist at the time, and number two, they cycle with the cell cycle. And the important thing is that they disappear and they come back. And if you stop them coming back by inhibiting protein synthesis, then the eggs don't divide. So it very much looks as though these are somehow intimately connected with MPF. I mean, we didn't really expect it to be MPF itself. We thought more likely it was perhaps an interconverting enzyme that gave rise to MPF. So I suddenly realized that I had stumbled on the secret of cell division, and I knew nothing about cell division or the cell cycle, so I had to start reading and talking to people about that. And DNA really is at the heart of this business. Sidney Brenner, when he saw the famous Watson and Crick model, said that DNA, it was a moment of absolute enlightenment, the reduction of biology to one dimension, because he realized that here in DNA was the equivalent of von Neumann's tape. Von Neumann was a very important computer scientist who talked about the problem of inventing automata, which self-replicating automata, and he realized that you had to have an instructional tape, but he also realized that the instructional tape had to be made by the automaton. So the automaton had to be able to make a new copy of the tape and be able to insert it into a new version of itself, so that the new version of itself would continue this process. And that's what life is all about, and that's really what DNA is all about. It's just the instructions. I mean, if you're a human being, it's all the instructions you need to make a human being. Or if you're an earthworm, to make an earthworm, or a bacterium to make a bacterium. We all use DNA in exactly the same way. And the key to putting a new copy of DNA in every single cell in your body is the cell cycle. Basically, what you have to do is you first have to copy the copy of the DNA that you've inherited, to make 2 copies of it, and then you have to distribute those copies to the 2 daughter cells. And the fact that DNA has 2 strands really explains why cells divide in 2. And the whole process is very nicely coordinated. And there are 3 very important switches in that process, points of no return. They are getting into mitosis, getting out of mitosis, and starting DNA replication. And they all have very similar characteristics, namely that the decision to initiate these processes is taken very carefully. But once you've committed to any of them, you can't go back. You can only go on. So that too was a very interesting conundrum. How could you make these irreversible switches out of systems of enzymes and their substrates? And it's taken, I suppose, from about, we should say, 1988 to the present day, and we're still not quite, haven't quite solved all the details yet. But there was a very exciting period in time when what had previously seemed deeply mysterious started to get worked out, because people could see the outlines. And the outlines really came originally from 2 kinds of research. One was, as I've described, these biochemical studies on frog egg extracts, and the other was from yeast genetics. And Lee Hartwell, pictured here, was really the hero, and he discovered that there were mutations which blocked cell cycle transition, which blocked these switches from flipping. And but because he was such a pioneer, he could never figure out how these genes actually worked, what they were, because they didn't look like anything else. But he worked out the logic of the cycle. And then, following in his footsteps, Paul Nurse, using a different kind of a yeast, made very important discoveries, focusing his attention on a gene called CDC2. And the reason why he focused on CDC2 was because it was a gene which, if you knocked it out, these yeast could not divide. They could grow, but they couldn't divide. And there was one particular mutation in this gene which didn't knock out activity, but made it more active, so that it actually accelerated cell division. And then it turned out that this gene, CDC2, was the same as one of Hartwell's genes called CDC28. And mysteriously, CDC28 didn't affect entry into mitosis. It affected entry into S phase. So that was very peculiar and caused some confusion at the time. And then Nurse discovered that not only did yeast have these genes, but humans had these genes. And so we realized that studying yeast genetics was a good way of getting at underlying problems of how human cells worked too. But these yeast genetic experiments didn't tell you, nowhere revealed about disappearing proteins like cyclin, because yeast genetics just tells you the components. It doesn't tell you how the components work. And so it was some time before we figured out the relationship between CDC2, CDC28, and cyclins, and it turned out to be a very intimate one. So going back to frog egg extracts, it turned out just as you can lyse reticulocytes and have an active protein synthesis system, you can break open frog oocytes and have an amazing system which is capable of catalyzing cell division, although there are no cells anymore. And this rich concentrated cytoplasmic extract, which you make simply by spinning these eggs. If you add nuclei to them, they form these beautiful meiotic spindles with purple chromosomes lined up between two spindle poles. And using this system, Jim Muller and Fred Locher were able to purify MPF, and to cut a long story short, it turned out that MPF was a complex of two proteins, CDC2 and cyclin B, our protein. So suddenly all was revealed, and the two together make once again a protein kinase, and then that suddenly became obvious how cell cycles were controlled. You activate a protein kinase, the protein kinase phosphorylates proteins in the cell, that causes their activity to change, so you bring about all these amazing things like the breakdown of the nuclear envelope and the assembly of the mitotic spindle, the condensation of chromosomes, and so forth. Many of these details still remain to be worked out. So what had previously seemed utterly mysterious suddenly turned out to be very simple, that the cell makes cyclin, the cyclin combines with the CDC2 is activated, it phosphorylates proteins, you enter mitosis, and then the cyclin is degraded, which turns off the CDC2, and you exit mitosis. Well, that was okay for a while, but obviously you'd really like to know how many proteins get phosphorylated by how much in order to bring about these changes in the cell's properties. And this sort of brings about an interesting conundrum, because if you imagine a cell that's not dividing, it's in interphase, and then it starts to divide, you phosphorylate lots of protein. To get back to where you were, all those phosphates have to come off, so there must be enzymes that remove the phosphates. And for a while it was really rather controversial about who did that, and there was no evidence that these things were regulated. And moreover, just to confuse matters further, the geneticists all said that protein phosphatase 1 was involved, and the biochemists all said that protein phosphatase 2 was the important enzyme. And there were, I can remember sort of fights at meetings when said, and it was most unedifying. Of course, the real answer is that both are required, and genetics tend to focus on PP1, whereas biochemistry tends to focus on PP2. And I began to get very interested in this. In fact, I was once asked to give a talk at a protein phosphatase meeting, and I said that I thought that probably phosphatases must be regulated in mitosis, because otherwise you couldn't completely phosphorylate things when you needed to phosphorylate them. And this was a thing that goes back to very simple biochemistry, namely the existence of things called futile cycles. If you have a process which sticks a phosphate onto one protein and an enzyme that removes that phosphate, it's obvious that you can't fully phosphorylate that protein if both enzymes are active at the same time. You would think there ought to be a control. If you want to fully phosphorylate things, you need to shut off the phosphatases, and if you want to fully dephosphorylate them, you need to shut off the kinases, and there needs to be a reciprocal relationship between the two. I mean, it's very obvious. It's just like filling a sink with water. You have to turn on the tap, of course, to fill a sink with water, but in order to actually fill the sink, you have to put the plug in, because if you don't put the plug in, the water just flows out through the plug hole. And if you think of the tap as the kinase and the plug hole as the plug as the phosphatase, that's exactly how to think about it. And the key person here who joined my lab late in life was a Japanese called Satoru Mochida. And between us we had a very happy, again, collaboration, and we discovered that indeed there is a phosphatase which is regulated, and this is the sort of thinking behind this is shown on this slide here. I mean, we know the kinase is regulated, but is the phosphatase regulated? And the answer is yes, it is. And again, to cut a long story short, it turned out that the regulation of the phosphatase is almost as complicated as the regulation of the kinase, and it's very important. And if you don't, it involves an enzyme called Great Wall kinase, another protein kinase. So I think the message is that protein kinases are tremendously important in regulating cellular processes, and I won't go into all the details, it's complicated. And the other interesting thing about this is a relatively simple diagram, and I learned an awful lot from a friend called Bela Novak, who analyzes these things in quantitative detail and builds models of how these control systems actually work. And together with Satoru, he was able to really get a very accurate quantitative model of how these kinases and phosphatases are mutually regulated. And it's a good example, I think, of how you actually do need mathematical, careful analysis and based on quantitative data to figure all these things out. So and I really can't emphasize too strongly how important it is to have people you like working with, and how important it is to keep your eyes open and think about things which you're not necessarily working on at the time in order to make progress. Because the nature of scientific discovery is, of course, you discover unexpected things. I think people are apt to think that scientific discovery is something like building a bridge. The principles are already known already, and it's just a matter of working out how many girders you need and how many nuts and bolts and things, and you just put them together. But it isn't like that. In biology, there are still a lot of things we really do not understand. I think I was lucky to be born when I was, in a period when we really understood so little, so that almost any stone you turned over had something interesting underneath it when you looked closely. But it still remains the case that I think the road may look straight immediately after where you are now, but over the crest of the hill it might turn to the left or to the right or peter out. And it's your job just to keep putting one foot in front of the other and keep going, just to follow the road wherever it leads. Thanks very much for listening.